Hi, my name is Christiane Funk, and I'm here to walk you through my poster on sex pheromone biomimetics for pest control and mechanistic probing of OPHR-PBB2. OPHR-PBB2 refers to Estrenia furnicalis pheromone binding protein 2. Estrenia furnicalis is otherwise known as the Asian corn borer, a major agricultural pest native to Asia and the Pacific Islands, responsible for crop losses estimated at 6 to 9 million tons per year. Current pest control strategies use the natural pheromone, and we're interested in looking at a novel strategy, either finding an attractant or an inhibitor for improved pest control. So we did our target design using Wabena docking. So we first looked at the natural pheromone structure, which is a mixture of E and Z 12 tetradecaenol acetate. So based on what we saw from those docking studies, we started modifying components of the structure and comparing the output from Wabena. So the three key features that we were looking at were the lipophilic tail, specifically modifying size, branching, and electronic nature. The polar head group, which in the natural pheromone is an ester, which is broken down by antennal esterases. So we're looking at metabolically stable ester isosteres such as amides, triazoles, and oxidiazoles. And then the third feature we were looking at is the U-shaped center, which we observe in docking studies. So we were interested in seeing how a scaffold that kind of forces that U shape impacts binding, whether it reduces the rotational barrier to binding, if that's a significant change that we'll see. So the lipophilic tail derivatives are the first series that we did. So these have all been made and are in the process of having the binding studies done in Dr. Smita Mohanty's lab. So we're waiting on data from this series. But based on the docking studies, they're predicted to have pretty similar affinity compared to the natural pheromone. So the data we receive back from the binding assays will really kind of help us determine how good of a predictor this method is. The next series we're working on is the polar head group derivatives. And then based on the binding assay results that we get for these two series, we'll be able to modify our optimized targets, taking the best features and adding them to that U-shaped scaffold. And just from the sample targets we docked for the optimized series, these are predicted to have much higher binding affinity, which is promising. So another thing that we are looking to learn more about through this research is the binding mechanism of OPHR-PBB2. There are several other lepidopteran pheromone binding proteins that have been well studied and work as a pH-controlled conformational switch. However, OPHR-PBB2 appears to function differently. So we're interested to see by probing binding through different structural changes of the target, how that binding process actually works. So again, this work is in collaboration with the Mohanty Lab at Oklahoma State. And thank you for your time.